should close the door. Welcome to the Center for Cardiovascular Regeneration at Houston Methodist Research Institute. I'm John Cooke. I'm chair of the Department of Cardiovascular Sciences, and I'm here with my colleagues for our weekly research seminar. And so today what we're going to do is I'm going to provide an overview of some of the work that we're doing, and then you're going to hear from one of the postdocs, Dr. Shu Meng, who will give an update on her most recent work. So thank you for joining us, and we'll go ahead and get started. This first slide is the title of the presentation, Nuclear Reprogramming, Role of Innate Immunity. And what you see there is also the Research Institute on the screen, and we're up on the 10th floor. Here on the 10th floor, we have a program in regenerative medicine, in cardiovascular regeneration, neuroregeneration, urological regeneration, orthopedic regeneration. So let's go with the beginning of the first slide. The concept that I'm going to be talking about today is this idea that innate immune activation activates a series of events that increase cellular plasticity. So what you're looking at is a process that we have termed transformation. It begins with some pathogenic challenge or some damage that the cell can sense. The cell has pattern recognition receptors. Pathogen-associated molecular patterns can be detected by these receptors. Damage-associated molecular patterns can be detected by these receptors. So the cell can sense a challenge. The cell can sense a pathogen, it can sense damage, and that triggers through toll-like receptors, through RIG1, receptor for advanced glycosylation end products, that can trigger a series of events that has been well characterized that results in the activation of gene expression that causes the release of inflammatory cytokines. So that's very well known. But what we've discovered is these same pathways have another limb. And the same pathways activate changes in the expression of epigenetic modifiers, changes in the activity of epigenetic modifiers that increases the openness of the chromatin, puts the chromatin into an open configuration so that the cell can just reach back and pull out whatever it needs to survive and adapt. So the activation of toll-like receptors and these other pattern recognition receptors, in addition to causing release of inflammatory cytokines, will cause changes in the epigenetics that increases the epigenetic plasticity of the cell for some fluidity of cell phenotype, so that the cell can change its phenotype to adapt to the damage or to the pathogen. So let me talk about the evidence that led to this concept that we term transformation. Well, pluripotent stem cells are a hot new area in medicine and science. When Shinya Yamanaka showed that you could induce pluripotency in somatic cells, this is a very exciting concept that led to the Nobel Prize for him in 2012. The idea that you can take a somatic cell like a fibroblast, and by overexpressing 4 genes, OCT4, SOX2, KLA4, c-MYC, you can change that fibroblast into a pluripotent stem cell that can become anything. It can become any cell in the body, endoderm, mesoderm, ectoderm, and the iPSCs now are being regularly generated around the world in research laboratories to understand pathobiology of disease and for screening of new drugs. iPS-derived cells, iPS-derived progenitors are being used also in the development of new cellular therapies. So because we're interested in vascular disease and cardiac disease in our Center for Cardiovascular Regeneration, we were interested in changing these iPSCs into cardiovascular cells to study pathobiology and also possibly for therapies. And particularly, we're interested in vascular therapies. So we started generating iPSCs, induced pluripotent stem cells, from fibroblasts, from human fibroblasts. and changing these human iPSCs into vascular cells, into endothelial cells, with the idea that we could potentially use these for therapy. Now, Shinya Yamanaka had used retroviral vectors to force the expression of these genes in somatic cells. But we wanted to avoid an integrating vector like that. We wanted to avoid altering the genome of the patient's cells. So we embarked upon an approach, a non-integrating strategy, to generate induced pluripotent stem cells. This was our idea, that we could generate the Yamanaka factors as proteins, as cell-permeant proteins. So basically we made a chimeric protein of each of the Yamanaka factors. So we generated OCT4, SOX2, KLF4, c-MYC. So we generated these transactivating proteins, these transcriptional proteins that activate the whole core pluripotency network. And then we, to those proteins, there was a polyarginine tail, which allowed for transduction across the cell membrane. So these proteins were Yamanaka factors that were able to get across the cell membrane. We demonstrated that these factors, Jun Lee, shown in the picture there, second from the top, she showed that these transactivating proteins could actually bind to their consensus sequence and in vitro. Johannes showed that the factors could get into the cell, and then it was the task of Arwen Hunter to find the right dose and the right duration, the right protocol to get the induced pluripotent stem cells. And at the very bottom, my Stanford colleague, Rene Rey-Opera, was helping us to characterize these cells as iPSCs. Well, this approach failed. And that failure led to the Cell paper that I'm going to talk about. Johannes showed that the factors could get. So this paper was published in 2012. Jun Lee was the first author. And the paper basically documented that nuclear reprogramming requires activation of innate immunity. So nuclear reprogramming, changing a human fibroblast to a human induced pluripotent stem cell, requires activation of innate immunity. Whether you're using a retroviral vector, whether you're using messenger RNA, whether you're using cell-permeant peptides, it turns out that the retroviral vector that Yamanaka was using to induce pluripotency was more than just a vehicle for the Yamanaka factors. It turns out that it had a very important effect on the cell that allowed the Yamanaka factors to work. So activation of these pattern recognition receptors like toll-like receptor 3 by the retroviral vehicle was necessary for this epigenetic plasticity that allowed the Yamanaka factors, OCT4, SOX2, KLF4, and c-MYC to actually work. So you need both. You need the epigenetic plasticity and you need the transcriptional factors that provide directionality to get reprogramming. And this was actually the key experiment. We were struggling to get these cell-permeant peptides to work. So Jun Lee went back to the beginning and said, okay, I'm going to compare one of these cell-permeant peptides, SOX2, in the first panel up there on the left, cell-permeant SOX2 in blue versus the retroviral SOX2 in red. And I'm going to look at all the downstream genes that are supposed to be activated to induce pluripotency. So SOX2, like the other Yamanaka factors, induces the expression of a cohort of genes that are required for the cell to become pluripotent. And NANOG is one of those genes that are downstream of SOX2. So it's something that you'd expect to increase in its expression as you go from a fibroblast to a pluripotent stem cell, because NANOG is required for pluripotency. So this was not the only one she looked at. We're just showing you here NANOG because it was basically exemplary of the downstream genes. They all had this kind of pattern, where in red, the retroviral SOX2 could increase the expression of NANOG. You see its expression increasing over 48 hours by 8-fold. Whereas the cell-permeant SOX2, even though it was getting into the fibroblast, the human fibroblast, it wasn't doing anything. So we knew that the proteins were getting into the cells, but they didn't seem to be able to activate the downstream pluripotency network. And this pattern that you're seeing in the upper left panel was duplicated using the other proteins, other cell-permeant proteins. On the upper right-hand panel, this is the work of another postdoc in the laboratory looking at cell-permeant OCT4 versus retroviral OCT4. So again, retroviral OCT4 is capable of activating an important downstream gene, a gene that's required for pluripotency, NANOG. But the cell-permeant OCT4, even though it's getting into the cell, we were able to demonstrate that it was in the cell, it could not activate the downstream genes. And we carried this experiment out even further, looking out at 21 days at gene expression, and you can see cell-permeant OCT4 isn't doing very much, whereas the retroviral OCT4 is increasing NANOG expression about 50-fold by 21 days, at which time we're starting to see the pluripotent colonies pop up. So the fibroblasts have actually now transformed into pluripotent colonies using the retroviral factors, but using the cell-permeant factors, we're not seeing any colonies. And so the question was, what is responsible for this difference? The cell-permeant peptides are getting in. We know from the in vitro studies that they could bind to their consensus sequence. In fact, we actually had done some work where we had knocked out one of these genes, like SOX2, in an iPSC colony. When you do that, when you knock out one of these core pluripotency genes, the iPSC colony will start to differentiate. It will differentiate into somatic cells. So you lose that pluripotency nature of the colony. But if we added the cell-permeant SOX2 back, we could rescue those colonies. So we knew the proteins could work. They got into the cells, they bound to the consensus sequence, they could activate the downstream genes, but it wasn't working in the fibroblasts. And so it occurred to me that maybe the retroviral vector itself had some activity. So I asked the guys to try just putting in a retroviral vector that's irrelevant, a GFP containing encoding retroviral vector. And that was the key idea that led to the finding that the retroviral vector was actually having some effect, because the retroviral vector here, together with cell-permeant peptide, is now shown in green, was capable of fully activating the downstream genes. So there's something about that retroviral vector that was having an important effect. And this idea had come to me because I had worked years earlier with Ed Makarsky on the effects of cytomegalovirus on endothelial cells. And I gave Ed a call to tell him about what we had found and asked for his help to investigate this further. And Ed talked us through the experimental protocols for determining the pathways that were involved in this activation of epigenetic plasticity. So essentially what we found is that the double-stranded RNA of the retroviral vector was activating TLR3 receptors to trigger downstream signaling, including NF-kappa B and IRF3, to cause global changes in genes regulating epigenetic plasticity. So that's the bottom line. And here's some of the data to support that. When we knocked out elements of the TLR3 pathway, when we knocked out TRIF, when we knocked out TLR3 in human fibroblasts, we were not able to get good colonies. What you're seeing here in B in the upper left panel at day 30 is an iPSC colony. This is a colony that's composed of about, several hundred iPS, induced pluripotent stem cells, closely packed together. Individually, these cells are round. They have a very large nucleus. They're distinctly different from the parental fibroblasts from which they were derived. So we had treated the fibroblasts 30 days earlier with the retroviral vectors encoding the Yamanaka factors, OCT4, SOX2, KLF4, and c-MYC. And then 30 days later, we get these nice colonies. Well, the scrambled control, there was no adverse effect on the fibroblasts. We could get the colonies with the Yamanaka factors. Mighty 88 knockdown, which knocks out an element of TLR, actually knocks out all of the TLR pathways except for TLR3. That had no impact. But if we knocked out TRIF or TLR3, we had reduced colony formation dramatically. And ultimately, I won't go through all of the data, but ultimately we were able to show that what was going on here was that the retroviral vector, by triggering innate immune mechanisms, was changing the chromatin from a tightly coiled, closed conformation that you see in the lower left corner to a more open configuration, which you see in the right panel at the bottom there. So chromatin, where it is locked down, where it was tightly coiled, spooled around the histone proteins, those genes are not being transcribed. It's in the open areas where the genes are being transcribed. So essentially what we found, without going into all of the data, is that the activation of innate immunity puts the chromatin to a higher probability of an open state on the right-hand side. And so, in summary, we call this pathway transformation. And essentially what happens when you provide a retroviral vector, you, in addition to providing the Yamanaka factors there, OCT4, SOX2, KLA4, c-MYC, those are important for providing directionality, for activating the downstream genes that are required for pluripotency, but you also have to activate the mechanisms of that increase epigenetic plasticity. So activation of innate immunity causes increase in P65, NF-kappa B, and IRF3 activation that leads to global changes in epigenetic modifiers. So the whole HDAC family is downregulated. Histone deacetylase is downregulated. That's a suppressive epigenetic modifier. The histone acetyltransferases are upregulated. That is something that enhances the open state of the chromatin by acetylating the histone proteins. And essentially what you have is, in this process of transformation, is an opening up of the chromatin. Now, Polish Shanda in the laboratory is showing that there's a Goldilocks zone to activation of innate immunity and nuclear reprogramming. In other words, when we're reprogramming somatic cells to induce pluripotent stem cells, there's a requirement for activation of innate immunity, as shown here in the blue line by NF-kappa B activity with luciferase promoter. So as we increase activation of innate immunity in cells that we are reprogramming, we see more reprogramming up to a point. And then with excessive activation of innate immunity, cells are still alive, but we get less reprogramming. Here we're using doxycycline-inducible murine embryonic fibroblasts that contain a cassette of the Yamanaka factors. And if we knock down innate immunity with a P65 inhibitor, we see essentially no colonies 3 weeks later. And with optimal activation of innate immunity, in this case we add a TLR3 agonist, poly-IC, we get a marked increase in colony formation. But if we overly activate innate immunity, there's a reduction in the colonies that we're getting. So what we're trying to understand now is this Goldilocks zone. What is different here from the other two zones? We believe there may be factors that are constraining epigenetic plasticity with excessive activation of innate immunity. And you know, indeed, when in our patients, we see people that have excessive activation, excessive inflammation that don't heal, like the diabetics with the diabetic foot ulcer. They have a ring of inflammation around the wound, and that wound is not going to heal. That ulcer on their foot is not going to heal until the surgeon does a debridement and removes some of that inflamed tissue. Similarly, we see in our patients when there's a impairment of inflammation. For example, with steroid treatment, we see an impairment of healing. So we think that this Goldilocks zone is not only important for nuclear reprogramming, but it may be important for healing as well. For regeneration, generally there may be an optimal zone of activation of innate immunity. And we're trying with our bioinformatics colleagues here to understand what are the genes that are involved in characterizing this zone. Well, now that we knew how to increase epigenetic plasticity, we thought about the possibility that we could manipulate this pathway, we could manipulate innate immunity, and then for therapeutic purposes. In other words, put the cells into a state of epigenetic plasticity and then provide the signals to get the cells that we need, what we call therapeutic transdifferentiation. The idea was to activate innate immunity and then to provide some cues. In this case, we're trying to get fibroblasts, human fibroblasts, to change into human endothelial cells. And that approach was successful. We've got proof of concept that was published last year in Circulation. And shown here is the protocol that we used up in the upper panel A. We activate innate immunity for about a week with poly-IC, the TLR3 agonist. And then we just provided factors that we felt might enhance endothelial phenotype. And those factors were BMP4 and VEGF and basic fibroblast growth factor. And we added 8-bromocyclic AMP. And we also added an inhibitor of TGF-beta receptors to prevent the reverse reaction, to prevent endothelial to mesenchyme transition. And we were able, with this protocol that we had basically empirically developed, we were able to, at some point, get endothelial cells using this protocol. And you'll see that if in the FACS analysis, if we don't add the TLR3 agonist, we just don't get endothelial cells. If we do everything else, we add the growth factors, we won't get endothelial cells. We actually have to activate innate immunity to get endothelial cells. Now, we don't get very many, but this is like the Wright brothers taking off and going 200 feet. We got, in this case, 2% of the fibroblasts to become endothelial cells. But they are endothelial cells, as shown by the heat map comparing the fibroblasts, the parental fibroblasts in this column, to the induced endothelial cells, two sets of induced endothelial cells, and human microvascular endothelial cells. You can see that the induced endothelial cells, these transdifferentiated cells, they were fibroblasts. We've transformed them into endothelial cells. They really look a lot more like endothelial cells, human microvascular endothelial cells, than they do parental fibroblasts. And we looked at the function, too. So these induced endothelial cells express all the markers that you'd expect them to express, CD31. They take up acetylated LDL cholesterol. They form tubes in matrigel. Fibroblasts don't do that. Fibroblasts don't do any of those things. This is my favorite experiment. This is NO generation. Endothelial cells generate nitric oxide. Fibroblasts do not. And you can see our induced endothelial cells generate a fair amount of nitric oxide under baseline conditions, which could be increased with stimulation with acetylcholine or calcium ionophore to stimulate the endothelial cells to make a little bit more, and very much resembled the human microvascular endothelial cells. And the fibroblasts, this is just noise. The fibroblasts don't make any nitric oxide. So the therapeutic potential of these induced endothelial cells was shown in our model of peripheral arterial disease. So human peripheral arterial disease is very common in the United States. There's at least 8 million, I suspect more, 12 million people with peripheral arterial disease that have narrowing of their limb arteries that leads to pain. Claudication is what we call it, intermittent claudication, limping with walking due to the pain in their muscles. They're not getting enough blood flow because of the narrowing of their peripheral arteries. And in severe cases, in about 750,000 individuals in this country, it leads to something called critical limb ischemia, which causes gangrene and limb loss. And so we investigated the therapeutic utility of these induced endothelial cells in this model of human peripheral arterial disease. It's a mouse model, mouse hind limb ischemia, where we tie off the femoral artery, the limb artery, and these animals will get gangrene to a certain extent. The blood flow is reduced to about 20% of normal after we tie off the femoral artery in all three groups. You'll see there's some spontaneous improvement in those animals that were treated with the vehicle, which was just the endothelial growth medium that we were using for our cells. But the cells themselves enhanced the recovery, enhanced perfusion. This is limb perfusion by comparison to the unoperated limb. And now we're getting about 60% of normal flow with the induced endothelial cells injected into the ischemic hind limb, similar to the genuine human endothelial cells. In fact, if anything, our induced endothelial cells seem to be somewhat better at improving perfusion in this model, almost getting back to baseline levels of flow. And here is shown the capillary density, an increase in capillary density with the injection of the induced endothelial cells or the human microvascular endothelial cells. And I should say here that this increase in capillary density in the ischemic hind limb after the injection of the cells is not due to the cells themselves incorporating into the microvasculature. We've sectioned the tissues. We don't see the human endothelial cells 2 weeks after injection. They're pretty much gone. But we have demonstrated that these induced endothelial cells generate angiogenic cytokines, and those angiogenic cytokines presumably are responsible for the increase in capillary density, which is totally due to an expansion of the murine microvasculature and not really due to incorporation of the human cells into the tissue. This is a hind limb ischemia score. The amount of gangrene was reduced. The amount of toe necrosis was reduced by the cellular treatment. So in conclusion for this part of the presentation, I wanted to introduce this concept to you today of transflammation and nuclear reprogramming. So when a cell is confronted by a pathogen, when it's confronted by virus or bacteria or by damage, there are molecular patterns that are presented to the cell that are recognized by the cell. So these pathogen-associated molecular patterns, these damage-associated molecular patterns, stimulate pattern recognition receptors on the cell, like the toll-like receptor family, such as RIG1, such as the RAGE, receptor for advanced glycosylation end products. These receptors can detect these patterns and triggers well-characterized inflammatory pathways that lead to the release of inflammatory cytokines, but also cause changes in the expression of epigenetic modifiers. I mentioned the whole HDAC family is downregulated. The histone acetyltransferase family is upregulated. These changes in the expression of epigenetic modifiers increases epigenetic plasticity and allows for a fluidity of cell phenotype that provides for some plasticity of the tissue. Now, we've made more progress in this area, and I'm going to introduce to you Shu Meng, who's our pioneer postdoc here in the Center for Cardiovascular Degeneration. She was the first to arrive. Actually, she arrived before me in June of 2013. And she's going to talk about the latest development in this field, which is the role of inducible NO synthase and its role in the innate immune response. So I'm going to, are you ready to go, Shu? Are you ready to present? So while we're switching over to Shu's presentation, let me just say that we've also, we won't be talking about it today, but it turns out that there are not only changes in NO generation by the cell, which Shu's going to talk about, that play a role in this process, but there's also changes in other free radicals like superoxide anion. So Shu's going to be focusing now on the role of iNOS and NO generation in this innate immune response and transflammation. So there's, in addition to changes in gene expression, there are changes in nitrosylation of the epigenetic modifiers that causes changes in their activity that lead to epigenetic plasticity. So Shu?
Thank you, Dr. Cooke, for a nice introduction. So today I'm also I'd like to present my work progress titled Transdifferentiation Requires INAS Activation, the Role of Ring1A as Nitrosylation. Well, has Dr. Cooke?
So the drug can leach out into the vessel. I mean, there's some ingenious things that work here. This is the Leaders Free trial published last year.
So the next question we ask is that what is the downstream target of amp-kappa B that modulate epigenetic plasticity in transdifferentiation? As we know that we already found the innate immune activation and the amp-kappa B signaling contributes to epigenetic plasticity, both in reprogramming to iPSC cells, but also in transdifferentiation of fibroblasts to endothelial cells. As going through the literature, we know that the inducible nitric oxide synthesis is a well-established downstream target of innate immune signaling and of amp-kappa B. And actually, INAS promoters has amp-kappa B binding sites. So, and also what we know is the inducible nitric oxide synthesis could generate reactive nitrogen species, including nitric oxide and peroxide nitride. Interestingly, this nitric oxide is a highly diffusible molecule and that can directly go into the nuclear and then post-modify proteins through directly binding to the sulfur group of the cysteine residue. And thus, we hypothesized that the innate immune signaling could go through the amp-kappa B, INAS, and NO signaling to directly post-modification of epigenetic modifiers, such as PRC complex and HDAX, to open the epigenetic, to increase the epigenetic plasticity by open the chromatin configuration. And so together with lineage specifications, this could contribute to transdifferentiation of fibroblasts to endothelial cells. Then the first question we ask is, does INAS get activated or upregulated during transdifferentiation? So the first experiment, we have repeated the transdifferentiation experiments by inducing human BG fibroblast through this 28-day protocol, and we can obtain 2% of CD31 positive IECs. And also, we have collected RNAs at different time points of this transdifferentiation process and examined the three types of NAS, the INAS, ENAS, and NNAS. And what we found that INAS is the only isotype that existed in the early stages of BG fibroblast during poly-IC stimulation. And you can see that ENAS is only upregulated during the end stage when transdifferentiation has happened, and we cannot detect NAS in this system. So this data suggests that INAS is definitely responsible to poly-IC stimulation and is potentially play a critical role in transdifferentiation. So INAS is upregulated. The next question we ask is whether INAS contributes to NO generation during this transdifferentiation process. So as shown in panel A, we can see that with poly-IC treatment for 3 days, using a DF-FM diacetate staining, we can see a significantly upregulation of NO generation. this chemical is non-fluorescent and only become fluorescent when it's react with nitric oxide. And also we use inhibitors to co-treatment with poly-IC. So we use inhibitors of alpha-kappa B such as B11, selastrol, and dexamethasone. And also we use PAN inhibitors for NAS, L-name, and also INAS-specific inhibitors, SMT and BYK. So our data suggests that both NO inhibitors and alpha-kappa B inhibitors could block the NO generation during poly-IC stimulation and transdifferentiation. So this data suggests that the NO is generated during transdifferentiation, especially through the other stages by poly-IC stimulation. And this process could be blocked by NO and alpha-kappa B inhibitors, suggesting that alpha-kappa B and INAS signaling is upstream of NO generation. So the previous data suggests that the INAS and NO is upregulated or it's associated with the transdifferentiation phenomenon. And the next question we ask is whether a NO, alpha-kappa B and NO pathway does contribute to transdifferentiation. So to do that, we actually use similar procedures. We go through the BG fibroblast through the transdifferentiation protocol, and we also use the NAS and alpha-kappa B inhibitors to block NAS and alpha-kappa B. And we observed the transdifferentiation efficiency by examining the CD31 positive IEC efficiency. And as shown here in panel A, that in normal transdifferentiation protocol, we can obtain about 2% of IECs. And together with alpha-kappa B inhibitors and NAS inhibitors, and also scavengers of the peroxide nitrate, these all inhibitors could block the transdifferentiation process, suggesting that the alpha-kappa B, NAS, and NO signaling is upstream of transdifferentiation. And further, as shown in figure in panel D, that poly-IC does induce INAS gene expression, while this process is blocked by alpha-kappa B inhibitor B11, suggesting that alpha-kappa B is upstream of INAS gene upregulation. So in previous data, we have used the pharmacological inhibitors, and the next question we want to use genetic knockdown and knockout method to test whether INAS is critical for this transdifferentiation process. So first, we isolated mouse embryonic fibroblast from either wild type or INAS knockout mice, and subject them to the transdifferentiation. And as usually, we can obtain about 4% of IECs in wild type mice, and this process was completely abolished by INAS knockout, suggesting that INAS itself is very critical for this transdifferentiation. And as a proof of concept, we can see the INAS gene expression is upregulated through the transdifferentiation protocol, while the INAS knockout mice, we cannot detect INAS level. And further, we also use the knockdown method. And in BG fibroblast, we have knocked down INAS using the siRNAs, and we found that the siRNAs could transiently knock down INAS. And we also use a plasmid to overexpress INAS and restore the INAS expression in INAS knockdown situation. And also we subject these groups to transdifferentiation. What we found is that INAS knockdown significantly diminished the transdifferentiation efficiency, while the overexpression of INAS that restored the INAS expression also restored the transdifferentiation process, suggesting that INAS is the key molecule that is responsible for transdifferentiation. So by far, we have proved that the NF-kappa B, INAS, and NO pathway is critical for transdifferentiation. So the next question is whether any epigenetic modifiers could be directly S-nitrosylated during transdifferentiation. As reported in literature, several key epigenetic modifiers are known to be S-nitrosylated, and some has been screened in vitro system. By Palash and Dr. Chen, our bioinformatician's work, we also found that 20% of the epigenetic modifiers could have the consensus sequence for NS binding. So following that, we have screened several key epigenetic modifiers in the transdifferential system. And the only gene that we found is this RIN1A, which is a critical component of the polycomb repressive complex 1. And what we found that is this gene is S-nitrosylated during transdifferentiation. So the first question we ask is whether the cellular S-nitrosylated protein level increase accordance with NS induction. And it did, as shown in panel A in Western blot, that with poly-IC treatment for 3 days, NS expression is significantly upregulated, together with a huge upregulation of the cellular S-nitrosylated proteins. And further, we performed co-IP studies to prove that RIN1A can be S-nitrosylated. And also, we found that this S-nitrosylation of RIN1A can be directly blocked by NF-kappa-B inhibitors, suggesting that NF-kappa-B induced NS is upstream of this RIN1A S-nitrosylation. And more interestingly, as shown in panel D, we performed co-IP studies of NS and RIN1A, and our data suggests that NS not only provide its product, nitric oxide, to RIN1A for its S-nitrosylation, but also it could directly associate with RIN1A. And we think this association may provide some specificity to its nitrosylation of certain cysteine site. So to further confirm the RIN1A S-nitrosylation and also to find the critical site that got S-nitrosylated, we have purified the RIN1A through immunoprecipitation and sent for mass spectrometry studies. And we found this 398 position cysteine is critical for RIN1A S-nitrosylation. So we further performed mutagenesis studies to mutant this cysteine, 398 position cysteine, into alanine to block this S-nitrosylation site. And as shown in figure C, that mutation of this position almost blocked the S-nitrosylation of this protein, suggesting that the cysteine 398 is the major S-nitrosylation site. And also we hypothesized that overexpression of this mutant form of RIN1A could block its reaction to poly-IC and block transdifferentiation. And as expected in figure D, when we overexpress wild-type RIN1A, the BG fibroblast still reacts to poly-IC and could transdifferentiate into IECs. But when we overexpress the mutant form, the C398 alanine mutant form, this reaction is completely abolished, suggesting that the RIN1A S-nitrosylation at its 398 cysteine site is critical for transdifferentiation. And further, we performed immunofluorescence study to detect INAS and RIN1A location during poly-IC and control conditions. And interestingly, what we found is that during poly-IC stimulation, INAS are majorly located in the perinuclear regions, and some of them would move into the nuclear. And also, we observed co-localization of INAS and RIN1A in the nucleus. These data suggesting, together with the co-IP studies we showed earlier, we conclude that INAS could co-localize with RIN1A during transdifferentiation and contributes to RIN1A S-nitrosylation and transdifferentiation. So the final question we ask is, how does this RIN1A S-nitrosylation contributes to epigenetic plasticity. As shown in Figure A, we found is that with polyacid treatment and transdifferentiation process, the INAS level is significantly upregulated, and this is associated with a global downregulation of H3K27 trimethylation level. And this downregulation can be blocked by AMP-kappa B inhibitors, A11. And also we performed the CHIP assay to examine the Ring1A binding to endothelial cell-specific gene promoters such as CD144. And what we found is that the Ring1A after S-nitrosylation binds much less to the endothelial-specific gene promoters. And together, the H3K27 trimethylation binding to this gene promoter is significantly downregulated, together with another repressive histone marker, H3 dimethylation. So altogether, our data suggests that the Ring1A nitrosylation is associated with decreased binding of Ring1A to chromatin and destabilize the PRC complex and subsequently decrease the repressive histone markers. So in summary, in this study, we found that the AMP-kappa B INAS and NO pathway is critical for transdifferentiation. INAS could directly bind and S-nitrosylate Ring1A through its 398 position cysteine, and this S-nitrosylation could decrease the Ring1A binding to chromatin, which subsequently decrease PRC complex stability and decrease the global level and also the specific H3K27 trimethylation level on endothelial cell-specific genes. All this contributes to epigenetic plasticity and pose the chromatin into an open configuration that facilitates transdifferentiation. Thank you.
Thank you, Shu. Nice job. So I think we have time for some questions now, and Dr. Von Bruno has the floor.
Sure, very nice data. You have actually looked into what pathways may be responsible for this epigenetic plasticity and transdifferentiation. The one question I have, you still have a very inefficient process. So you have 4.5% of your cells are responding and being, in spite of the fact that you're showing that a lot of them are expressing NO, that a lot of them are responding, and clearly this pathway is contributing. What is different between the population of non-responder and the population of responder? What do you think it's responsible for limiting these cells from transdifferentiating? Have you looked into gene expression? Do they respond different in where the chromatin is being modified? What is different about, is it cell cycle dependent? What is different about this population, and how would you look into that?
Yeah, that's a great question.
Well, I think that we really don't know. We don't know. It's a great question. What we need to do, I think, is some single-cell RNA-seq work. We need to understand at a baseline level if the fibroblasts themselves are different. I suspect that the cells are synchronized, right? The cells are synchronized, but that doesn't mean they're all the same genetically. So one of the things we have yet to do is to understand we need to do single-cell RNA-seq of the starting population, because the starting cells may be different. There may be cells within the population of fibroblasts on a dish that are just more likely to be reprogrammable. So that's one possibility. And the other thing... Address that?
I don't know whether I should talk about it.
We're trying to do some lineage tracing studies right now, but I think that your question is a good one. We just need to do some single-cell studies and find out if the starting population is different. If there's something that's different of the cells that reprogram, what is different about those cells that allows them to have that epigenetic plasticity. We're certainly missing something. Maybe, and the other thing is, maybe our protocol is not. This is an empirical protocol. We've tried to optimize it, but we've tried to optimize it empirically. And one of the things that we're searching for money for, we've written to NIH for funding to do a small molecule screen, image-based small molecule screen, to see if we can improve the protocol. So that's another issue as well.
Poly-IC is currently being used as an adjuvant for therapies for cancer, against cancer. So what is it that predispose these cells, so rapidly dividing cells, into these chemical agents? Have we mapped where in the chromatin you're having more susceptible spots and what genes are being responsive to this reprogramming or plasticity? That might be a way of assessing what genes are involved in this process more specifically. And just that will be a Kai-Fu, a Kai-Fu homework. I don't know if you want to add to that.
So I guess that's a very.
This is Kai-Fu Chen, our bioinformatician.
So I think in your cell paper you already have RNA-seq data for the fibroblast cell treated by poly-IC or not, right? We already have those RNA-seq data, and they already show that the epigenetic factors are regulated.
But would it be RNA-seq data, or would it be looking in the chromatin? Because RNA-seq suggests which RNA transcripts are being
differentially expressed, no? But not necessarily which sites, where in the chromatin you are causing a opening configuration as opposed to where in the chromatin you're inducing changes.
So I think that's the reason. I think in our last R1 grant application, we proposed to do, for example, ATAC-seq to profile open chromatin regions in a cell and how those open chromatin regions are regulated by the poly-IC treatment.
Can you tell us a little bit about ATAC-seq?
Yeah, basically, ATAC-seq is recently developed new technology to detect open chromatin regions across the whole genome. It's not only detect any specific region, but extensively detect almost each open chromatin region across the whole genome. And these open chromatin regions are the locations that, for example, the Yamakara transcription factors can bind to the chromatin. So if the chromatin is open, these transcription factors can bind them. If it's not open, these transcription factors are not able to bind the chromatin, and thus cannot, activate the transcription and so on.
Yeah, and also I think one thing I want to add is that, as we know, the in vitro system is a proof of concept, and also we want to show that the combined activation of innate immune and then combined of the endocellular developmental signaling could provide a nice environment for the fibroblasts into endocellular cells. And the more interesting, what we really want to look into is the in vivo system. And we want to see how fibroblasts in vivo could really contribute to endocellular cells. And I don't know whether we should talk about it.
Well, I think we can say, in closing the conference today, we can say that what we're hoping to do, now that we understand this process of transformation, is to use it in a therapeutic manner, because that's what we're trying to do here in this institute, in this center. We want to make fundamental observations, and we want to transfer those fundamental observations into something that's useful. So one of the things that we're working toward is a regenerative therapy based on transformation. So how can we modify the epigenetic, how can we increase epigenetic plasticity to improve cellular fluidity for healing? Here's an example. If we can change some of these fibroblasts that are forming in the area of an injury and releasing extracellular matrix to form a scar, if we can encourage some of those to become endothelial cells, those endothelial cells will self-assemble to form capillaries that will provide nutrition and will provide a niche for true tissue repair. So the idea is to improve tissue repair by enhancing this process, by modifying the scarring process and changing the scarring process in such a way that we get tissue. So that could be useful for wound healing, it could be useful for myocardial infarction, it could be useful for a lot of disorders that are characterized by some acute tissue damage and with a healing process that involves scarring. If we can modify the scarring process in a way that enhances tissue formation, then we can have true regeneration or better regenerative therapies. So thank you, Shu, for that latest data. Really exciting, and look forward to seeing more. And thanks to everyone who's viewing this, and there'll be more exciting presentations to follow. Thank you.